The second type of dealkylation enzyme is O6-alkyl-guanine DNA alkyl transferase 2. So this enzyme is another E. coli's alkyl transferring enzyme. It has properties very similar to that of C-terminal domain of O6-alkyl-guanine DNA alkyl transferase 1. It also transfers a single alkyl group from the O6 position in guanine or the O4 position in thymine to one of its own cysteine residues. So it behaves similar as the C-terminal domain of alkyl transferase 1 enzyme. After this transfer, the enzyme loses its activity and so is also classified as a suicide enzyme. However, this enzyme does not appear to be subjected to genetic regulation. It cannot be converted into transcriptional factors and cannot stimulate the transcription of its own gene. So it cannot regulate the synthesis of its own. Its function appears to be to protect bacteria from alkylation damage during the time that it takes for the gene of O6-alkyl-guanine DNA alkyl transferase 1 to be fully expressed. So there may be some time gap between the start of expression of the gene for the synthesis of alkyl transferase 1 enzyme to the end of this expression. So between this time, that alkyl transferase 2 enzyme that protects the DNA from alkylation damage. The third enzyme, dealkylation enzyme, is alkyl guanine DNA alkyl transferase, and this enzyme is a eukaryotic enzyme, and this protein removes alkyl group from O6 methyl guanine or O4 methyl thymine in similar to the bacterial O6 alkyl guanine DNA alkyl transferase 2. Human alkyl guanine DNA alkyl transferase is of considerable clinical interest because many therapeutic agents used to destroy cancer cells are alkylating agents. So that's why it is very significant clinically. When alkyl transferase activity is very high in the tumor cells, these chemotherapeutic agents are not effective because chemotherapeutic agents will cause alkylation, and this alkyl transferase activity will cause dealkylation. So these chemotherapeutic agents will not be very effective because their effect will be reversed by these enzymes. On the other hand, when alkyl transferase activity in the surrounding healthy cells is too low, when these enzymes are very less active, the chemotherapeutic agents will kill these cells. The human protein or enzyme is also important because it helps to protect the cells against carcinogenic alkylating agents, because such type of alkylations can convert normal cells to carcinogenic cells, and these enzymes protect the cells from being converted to carcinogenic cells. Another enzyme is ALK-B. This is entirely different type of alkylation damage repair activity, and it was found in E. coli in 2002, and this activity is carried out by a protein which is called ALK-B. ALK-B catalyzes the direct conversion of 1-methyladenine, 1-methylguanine, 3-methylcytosine and 3-methylthymine to the respective nitrogenous bases, that is adenine, guanine, cytosine and thymine. The ALK-B catalyzed reaction requires iron, molecular oxygen and alpha glutarate. So these three requirements are for the ALK-B to act. A similar type of enzyme has been found in other organisms including human and mammals. So here you can see the mechanism of action of these ALK-B enzymes. So this is 1-methyl adenine and this is alpha-ketoglutarate and this is converted into succinate, and here iron is present and ALK-B enzyme is also present, and after this intermediate this adenine has been restored and formaldehyde has been released. So similarly this is 3-methyl cytosine and it is converted into cytosine. So this is the action of ALK-B.